Today is our discussion about feed-forward neural networks. One of the first strong metaphors that was unifying brain parts with computational models was the idea of David Marr. You may or may not have heard of David Marr, M-A-R-R, who had suggested that the Purkinje cells in the cerebellum acted like perceptrons. We're going to talk about the perceptrons today. So it was one of the first metaphorical uses of a computational analogy to a cell of the brain to try to figure out what the work of the neural networks of the brain was. And yeah, the reason why I wanted to show this is a micrography from a friend of mine, and you can see here how neatly arranged the Purkinje cell somata are. If you might actually remember also that the arbors of the Purkinje cells, they're flat. I don't know if Josh told you that they're flat and they're opposed one on top of each other, and the parallel fibers cross them. So if this is a Purkinje cell, there are parallel fibers crossing perpendicularly to it. It's fun to think about this particular arrangement. It's unique. If you think about the pyramidal cells in the cerebral cortex, they have a radial symmetry. That means their dendrites go in all directions, and you cannot find a preferred orientation of the cell. The Purkinje cell is not like that. The Purkinje cell is very flat. Ultra flat. It has about, in a mouse, the size of the Purkinje cell would be 1/4 of a millimeter, about 15 micrometers, all of the dendritic tree, but the width of the Purkinje cell would be about 20, 30 micrometers. So it's basically a very thin brick, and this thin brick is being penetrated by these parallel fibers on the other direction. This has caused Marr to think, well, what is the Purkinje cell doing, right? I mean, it's looking at, say, 150,000 inputs and producing an output. And that output, whatever output it is some sort of combination between what happens between these inputs and what happens to the outputs. There are a couple of differences between what Marr intuited that the Purkinje cell would be doing and what the Purkinje cell is actually doing. But by and large, the metaphor sticks. And we're going to work through today a little bit more of our understanding of perceptrons and build on to deep networks. I now realize that in order to really discuss my background, I should probably have stopped the share so that you can see it bigger, in any case, I'll continue the share. Good morning, everyone. So the thing that we have for today, we're going to be discussing deep network architectures between today and tomorrow. So tomorrow we will have a time to work on this together. We're going to be using the so-called playground TensorFlow, which is ultimately it is a demonstrator for how deep neural networks are transforming input data into output data. And we will be discussing it in detail tomorrow. Today, I want to give you a bunch of intuitions related to the underworkings of these deep networks. Now, you talked about deep networks before, and now you probably have already simulated a little bit of the general idea. But the way that this lecture ties in with a lecture on Friday is that we talked about functional localization. And we talked about hierarchies, basically the idea that everything is a receptive field and they're being combined. I don't know, if this is the summary that you've taken from last Friday, but at least in the categorization of input patterns onto higher level percepts, our understanding is very close to the idea that hierarchies lead to invariant pattern recognition. How it does it, we're going to still discuss. I couldn't managed to do this lecture without bringing to your awareness the neocognitron. So the neocognitron was the first deep learning architecture by I think Satoru Fukushima is his last name, in the end of the 70s, because it is prescient. And even though it has, it smells of old, it carries all of the essential elements of modern deep learning. So I want to tell you a little bit about that. We're going to be watching a 7-minute video about basically a documentary on the 80s about the neocognitron. Then I want to tell you a bit more about convolutions in general and filters. So the idea that we're extracting features via filters from images has already been building up in our recent past. But now I'm going to go a little bit more into detail. What is a convolution function? What is a filter? And how do you apply one to the other? Sem, one of our TAs, he managed to put together a little Jupyter notebook on the topic of convolutions. It's not an entire full-fledged project, but it walks you through applying filters to convolutions, and I think that you will enjoy that. And then tying in with tomorrow, that we're going to be working together on this in groups on this playground TensorFlow. We're going to talk about classification, regression, perceptrons, and decision boundaries when they basically become more complex. And Elias at 11 is going to be joining us, I think 11:30. He was going to be joining us from the middle of his cognitive neuroscience course in Amsterdam to walk you through the nooks and crannies of the HEB project. You might have noticed, I mean, there were a couple of things we were still changing as you were working on it. So Elias is going to tell you why and what happened there. And he was going to go through some of your questions. Kate had a question about, how is it that it is extracting these regularities? Why is it that the network sees a certain pattern with each other more than other times? Maybe you remember your question, Kate, but we're going to come to that at 11:30, all right? Now, with 16 participants, I'm feeling fully comfortable to start. Any questions before we go? Now, this is something we're going to come back to a couple of times still in the next 2 weeks. We're going to be discussing more structure and functional relationships than we've ever done so far. One thing to get stuck from today, one of the learning goals, is that hierarchies are great for pattern recognition. I think that probably already got across last Friday. Did it? That hierarchies are good for pattern recognition? More about that. When we talk about recurrent neural networks, we're going to be talking about pattern completion, such as the Kanizsa triangle or other types of filling in with feedback excitation. We're still going to be talking about decision making and reinforcement learning. And so all of these, obviously we already did talk about motion perception and sensory motor control. But now with recurrent neural networks, we're going to get closer and closer to the notion that memories relate to attractor dynamics in neural networks. But for today, exclusively, we're looking into pattern recognition. And this is a bestiary of neural networks that I've shown to you before. And particularly, you will notice that by and large, networks complexify, but they still have, it's basically neurons. it's basically neurons that are sending input and having some sort of transfer function, and they're connected in different ways. You've asked not very long ago, what was the difference between a feed-forward network and a recurrent neural network? Now, you'd be able to tell by looking into this cheat sheet, if you like. We're going to be discussing the perceptron first. Then we're going to discuss feed-forward neural networks. But you may already remember that we did talk about radial basis functions, or in fact, networks that had as inputs neurons with different tuning curves. I don't know if you remember, I've mentioned what a radial basis function was before. Remember when you have like the cricket hair cells in the leg, so the 4 mechanoreceptors at the leg, they are effectively radial basis functions because they are sensitive to a certain interval of the input space. So if the input space is wind coming from 4 directions, they're summing to cover the entire space, is the word that we generally use. But by now you should be like more than able to tell the difference between feed-forward neural networks and recurrent neural networks. Here you see, can anyone tell me the hallmarks of these two? So in the recurrent neural network graph here, maybe one thing that is not coming across is that connections can also go backwards. So here there are no arrows going forwards and backwards in the diagram, but in recurrent neural networks, connections go not only feed forward, but also backwards. And they can also map onto the same neuron. So that's the essence of a recurrent neural network. But today we're going to talk about networks that go forward only, and what happens with the input that they're getting as they're transforming the input that way. So I have one single slide about hierarchies that is going to bring us back to what we had last week. So from this, feed-forward and radial basis functions look quite similar. They are in fact identical. The only trick is that for radial, thank you for your question in the chat, I don't know if you've noticed, the difference between the radial basis functions and the feed-forward neural networks is that the radial basis functions have inputs whose weights cover the input space in a specifically regular way. Basically, it tiles the input space. Tiles as in tiles in the bathroom, you know, they cover the input space. If the input space was the wall of your bathroom, the tiles would cover it. And that means that they have a receptive field only for when you touch that particular tile, only that tile will light up. So if you had a bathroom with LED tiles, and you touch the different tiles, they are receiving from a particular physical space and that they are covering, so that you can best reconstruct where you're touching the tile was. Poor metaphors as always. So good observation, Phong. The feed-forward and radial basis functions are very similar, and in fact, so is the deep feed-forward neural network with the neural network with a basis function, or even the deep convolutional neural network. We're going to get to this. We're going to talk about what these pink cells here are today as a function of filters to continue assembling from the knowledge that Josh brought us. Okay. Who can explain to me this slide? Can you see it with clarity? So there are many features of this slide. I mean, what do you observe here? So, start with, say, for example, let's start with the receptive field size. Okay, so I have my cursor here pointing receptive field size. What do you notice? So what is this graph trying to convey? What's this graph about? The left side, actually the right side too, but what's the left side of the slide about?
It's the pathway of like vision perception, I guess.
Very good. So does it relate to something that, how does it relate to what Josh told us last Friday?
Yeah, so when we start from retina, we start to collect like low level like processing going to the more like high level processing. So I guess in the first like a few steps at Greta 1000, we collect like more low level features. And then as we go more into like visual cortex to like the continued from ventral pathway, it becomes more like complex. So I think the features add up onto what the other like steps have already processed. So
Perfect.
it builds on top of that. Like
perfect. So excellent, John. So what do you see? So in the bottom, there seemed to be a split. In the bottom of the graph, there seems to be a split. Does anyone else recognize the split? Would you like to tell me what that split is about? I mean, clearly it's written on the slide, but even that is good. Someone else, Chenzo, thank you for opening your mic. So, yeah.
You have the temporal lobe and the dorsal lobe, and that divides to perceive what an image is and where a thing is in space. So that are two different kinds of pathways.
Yeah, so the distinction between the vertical and so the ventral and the dorsal pathway, or the in fact, like you said, temporal, more like
yeah, I'm sorry, I meant
cortex. That's perfect. I mean, it's not occipital, it's temporal and parietal. So parietal cortex on top. Lesions in the parietal cortex, for example, give you out-of-body experiences. So you're no longer in the space where you thought you were. You have illusions of space, right? Another thing that relates to the parietal cortex is the objects you can grasp. So in the top of the brain, where an object is represented, like you just pointed out. In the ventral pathway, you're distinguishing semantic information. It's kind of interesting to think about how is it that the split happens kind of late in the processing stream, and you do need these blocks before you're able to assemble, on one hand meaning, on the other hand a position, right? We still should be thinking about distributed processing. Now, what is going on in the right side column, receptive field size? Can anyone tell me what's going on here? Is it too small, like because I'm selecting a piece of my screen, or too low resolution? Make it grow a little bit. So what are those concentric lines? Let's start with something simple. Notice that unfortunately the picture seems to have like a relatively small resolution already to begin with, but I think it's mostly all right. So what are the concentric lines? The red concentric lines, if they appear red to you. Notice the title of the slide. Here's Deep Hierarchies in the Visual System. And where does the visual system start? In the retina. And what is the size of a receptive field? What is the size of the receptive field? It's a question in your test. Let me make it easier. What's the size of the visual receptive? What's the size of a visual receptive field?
Is that the fovea?
That's right. The amount of fovea that excites a single cell, or the amount of retina that excites a single cell. So in the retina, the receptive field is of the size of the photoreceptor. Because one photoreceptor is activated by the light inciting on it, right? So it's super high resolution because you have a lot of photoreceptors in the retina, but they're very, very tiny because they only receive light inciting on themselves. But then as you propagate a little bit upwards, you have slightly larger receptive field sizes. And also in this case, with the ganglion cells, a little bit sparser. And what happens with the so-called simple cells there? Where are we looking at? So these concentric lines now here in V1, what do they represent? I wish I had some drums here.
The receptive fields gradually get smaller when the orientation or the image is more complex.
That is certainly something. So first, the question is, what are the concentric lines? But yes, let's get there. What are the concentric lines to start with? I mean, according to what you said, you know it, Chansu.
Isn't that like the angles of like receptive fields? So the middle is the right at the fovea, and then it moves along with.
That's right. So that's the visual field. So this is the answer I've been searching for. So the concentric lines is basically a representation of the visual field as the visual field propagates through the different layers of the hierarchy. So part of the idea is that the retinotopy goes on to lateral geniculate nucleus and goes on to V1. So even in V1, we have a representation of the entire visual field. We talked about it before. So the idea that the retina basically topographically maps onto the visual cortex. And then what Ruud was actually pointing out, I wanted first us to compare it across the column. So in the column, the receptive field sites from the retina to the thalamus, it's grown a little bit in size because obviously in the retina, it's a single photoreceptor, and in the thalamus, there will be one plus all the surrounding ones. So it's already more cells you need. If you need more cells, then to do the same processing, you have to look into a slightly larger patch of the world. And if you want to find out what is the orientation of that selection of photoreceptors receive from, so, oriented bar, you will need to include even more space. So you cannot determine whether like this finger here is oriented horizontally unless you take a substantial amount of it into your network that is distinguishing, right? Like it's like your do-it-yourself receptive field. If I give you only one pixel, you can't ever tell what's this. You need more space. You need more area to aggregate that information from. Is that obvious? Now, what we're seeing here is also, as Ruud pointed out, further away from the fovea, the acuity decreases. The receptive fields get larger, because they have to include more of those sparser photoreceptors to make sense of the presence of a certain feature on the visual field. And as you go to V2, where you have like these much more complex objects, you need to integrate even larger patches at the level of the retina to be able to distinguish a texture. Is this obvious? So in V4, however, You have the disappearance of the distal receptive fields. Oh yeah, here. What is the sort of x-axis in the receptive field graphics? The x is the radial axis. It is the axis of the, so Kate, can anyone tell me what's the x-axis in the receptive field graphics? Chanzo already basically answered it, but I want to see if anyone else like now sees it. It represents a distance from.
Well, distance to the fovea, right?
It's from the center of the fovea. So here they could have represented the entire retina with an entire circle, but since it's going to be the same over any of the 4 quadrants, they just chose one quadrant to represent it. Now, this is our best knowledge about how hierarchies extract these features. And I think that it presents a very compelling image of how much we understand about feature extraction. There's a couple of things we don't understand, and we're going to get to them. But for the moment, this is good. Now, here on the right side of the slide, what do we get? What do you see? Today, I'm going to edit the silences from this lecture. Julian, you opened your microphone.
Yeah, it's like V1 and V2 need more receptive fields, or need more to understand what's going on in the picture.
What else do you see? So actually, these are from completely different papers. This is the canonical view of hierarchical processing in the visual strain. So what's the difference between the retina and V1 in that right size field? Jacobina, can you tell me what's the difference between V1 and the retina in the right side of the slide?
No, sorry.
If you look at the retina and V1 on the right side, like, do you see the yellow dots and the green dots?
Yes.
And what's the difference between the rectangle in the yellow and the rectangle in the green? What are little blue yellow dots representing, in your opinion? In the retina, for example. Can anyone come in and help?
I think the cells.
These are, yeah, neurons, pixels,
Yeah.
cells, exactly. And what's the difference between the retina layer and the V1 layer in this picture?
It's
Jacobini,
larger and it has more neurons.
Yeah, right? Jacobini, can you see that there are more neurons in green than in yellow? Are you surprised by the fact that the retina has less neurons than V1?
No, I'm not surprised by that.
No? So why are you not surprised? I ask you because clearly, you need to make, you want to make a picture in your brain, but you only have the neurons in your eyes to work with. So basically, that's all the information that is coming. Why do you need this expansion of the size in V1? Yime, Constantine, and Alessandro, anyone has an idea about this question? I beg the pardon of those that think that we're going slow here, but if we're not all on the same page, then I refuse to move forward.
Maybe V1 and V2 are bigger because, as you said, you need to see the bigger picture to find out what the orientation is, for example.
Yeah, exactly. So that is right. But I'm wondering if our silent members are able to see this. The important thing here is that in order to extract features, you need to combine like the inputs in different ways. And so you will definitely need more neurons for features that you want to extract. There is an expansion in size between the retina and V1 of about 10 times. And so I just want us to think a little bit about the contraction and expansion that there can be. So in this case, divergence, remember? One of the motifs that Josh kept on bringing back on Friday was the divergence onto the neurons, and the convergence onto some specific neurons that are doing the recognition. Now, the notion of invariant processing goes very well together with the notion of perceptual invariances, which is one word that comes often back when we're discussing the visual system. Not only the visual system, but any sensory system whose underworkings requires it to extract specific semantics from worldly objects. A banana tastes like a banana. How do you put a banana together? You have to take the different chemoreceptors in your nose and in your tongue, integrating them into an invariant percept of a banana so that you can, bite it and tell the other person it is a banana. They see it, so the subjectivity of processing disappears when you think about the necessities of the object. So the kinds of things that you would have to extract to be able to agree with a different person that is a banana. So the so-called overlap of intersubjectivities is actually objectivity. This is philosophically spoken. The idea is that if I overlap my subjectivity with yours, then we have a little bit of objectivity in it. Now, where is that objectivity coming from? Well, it's coming from the similarities in our neural processing, so that we are extracting things about which we can agree. The things that we, if you look at a ball, it is circular, it occupies your visual cortex in a specific way. That's why you can tell me it's a ball, and I can agree with you that it's a ball. Extracting those invariances is what these hierarchies were doing. And so what Josh presumably got across last week is that you have the combination of multiple different input fields so that you can assemble the objects. But there are many ways in which you can change the object. For example, you can make the ball smaller or larger. Is it still a ball? So it is still a ball because it's invariant with respect to size in your visual field, in terms of the semantics that you acquire. Proximity, is it close, it's far, it's still a ball. So you have transformations, close, far, rotation, that will keep certain essences of an object unchanged. Thinking about invariances is key to understanding what are features and what features would the neural system would like to extract. So in here, a cube, for example, is not rotationally invariant in the sense that it doesn't look the same from every single perspective you hold it. However, it's still a cube. And so there are some specificities of that three-dimensional object that, even though it might have like different orientations, it still will be perceived as that particular essence. How do you build essences? In any case, before we go forward, I just wanted to establish the notion of invariances. You've been talking about it when you're talking about receptive fields that prefer a grandmother. A grandmother is an invariant of a particular neural network processing if it responds to grandmother no matter what her orientation is. If the grandmother upside down, should still be grandmother, right? This is one of the fundamental problems of deep neural networks, and this is one of the first problems that deep neural networks has tackled. And Fukushima came up with the idea of neocognitron to try to make sense of that multi-layered organization, and how is it that these receptive fields would be extracting information. Now, before we go further, I have a quiz for you. Okay, so I sense you pit on the slow today, if you don't mind my saying. So I just told you that invariances, the essence is what remained unaffected by a transformation. And I want to think a little bit about, in the brain, you look at stuff and everything is like permeated with variability. Stuff is all over the place. But neuronal dendritic trees change, the neuronal ion channels change, and the neuronal structures change, and the neuronal cortical thickness changes, and your photoreceptors are different from your friend. And so there's 100 million, billion, trillion things that change. And we, however, have to assemble some sort of invariances from it. I wanted to think a bit about these different methods of recording the brain to look into the specific invariances, and then we're going to play a little game. So for example, you have here an invariant that appears as an activity as a function of a location. You have in Pinot invariances that appear with fear. We have some white matter tract invariances. We have some dendritic tree shape invariances. For example, here you see a dendritic branch with a few synapses protruding, and this is electron microscopy. If you go to across healthy brains, you will find the probability of encountering a spine or a synapse more or less the same. Whereas if you go into an Alzheimer's patient, you will see substantial changes in that statistical property. So there's invariance in number, in count, in density, even though there's no invariance in the wiggliness, like every dendrite's the same. In Sporns 2008, they measured hub capacity from different brains. And so these are different hub-like cells from diffusion tensor imaging. And in Tononi, they were measuring neural correlates of consciousness as coherence from EEG. So, invariance of behavior. The idea that you have something you measure that relates to something specific. Remember, we've talked now about a few invariant response cells. So these are invariant response cells. First, I want you to share with me the definitions of those cells. Can you tell me, one of you at a time, come in and just explain succinctly what each of these are and what is the invariant, if you can. Explain what it is and explain what is invariant.
Okay, so a mirror neuron is a neuron that starts firing when another animal or human does something, and your brain is expecting that you might want to or have to do it as well.
Indeed. So it's an invariant to an action. Would you agree?
Yeah.
So either observed or performed, it's the action that is invariant in the mirror neuron representation. Okay. So how about the grandmother cells? Can one explain succinctly what they are and tell me what are they invariant to?
I guess it reacts to something very specific, like your grandmother.
Invariant to grandmother, something very specific. An action could be very specific, but it's even more specific than that. It is a person, and it is an old person, and it has a certain familial relationship to you. You haven't talked about I'm not completely certain you have talked about canonical cells before. But the canonical cell is just like a mirror neuron, only that it doesn't require you looking at another person. So a canonical cell is an action cell that fires if you perform an action in the dark, if you perform an action in the light, or if you just think about performing the action. So they're also, so this is it's also an invariant to action with the detail that it does not fire if you're looking at someone else performing the action. You have to be the agent and agency. Okay, place cells.
Those are invariant to a place. So when you get to a certain place, they fire.
Yeah, so they are invariant to a specific location in the environment. I think many of you would be able to tell. This particular one, we've drilled them a bit. Do you remember where they are in the brain?
Was it the
entorhinal?
hippocampus?
Yeah, so entorhinal cortex of the hippocampus. So how about head direction cells? I don't know if you've been drilled on head direction cells as much as place cells, but I wanted to, so if you don't know, just give me an intuition. What are head direction cells about?
Probably the direction of the head or position.
Direction of the head with respect of the body or with respect to the world?
I think respect to the world.
Yes. So it is where your nose is pointing.
Yes, I think so.
And in fact, head direction cells are invariant to where your nose is pointing. Now border cells. When do the border cells fire? Well, festive, a cheap one would be, yeah, Nevin?
Yeah, it's border cells is when there's, for example, like right behind me, there's like the corner line, and it's so the one side is different to the other side, and that's the border in between.
Indeed, very much so. So when you're close to a border, your border cell will fire. It basically tells you how close you are to the rest of the environment. Think a little bit about the parietal cortex. And then odor cells, you can have a banana cell, which will fire very much when you see a banana and not as much when you see a carrot. So semantics crosses modalities. I am sure that when you came to the computing brain, you might have even thought about some of these things, how the brain encodes, like you might have read about place cells before. It's like, but surely the brain must be doing more than just recognizing places. Surely that cell fires to the place, but what else has been necessary before a cell could fire to a particular place? The discussion point here is that we give names to cells in neuroscience when we find the thing that they like, because the thing that they like is most likely a niche, ecologically, semantically valuable object. It's something that you can segment from the rest of the world. And you can say, okay, that's good. I mean, it's good if you know where you are. Now, how the networks produce that invariance is a different question. I just want you to appreciate a little bit the fact that we as neuroscientists name cells because they help us understand what is being processed. So whenever you look into one of these cells, the next question you should be thinking is, how do they come to be? How does a grandmother cell come to be? How does a mirror neuron come to be? How does a canonical cell? What are the preconditions for their emergence? You with me? Now, can we play a little game, and then you're gonna get a little bit of a pause. So in this little game, you will very quickly be presented to a slide, and you have to write in the chat what type of cell it is as fast as you can. There is sufficient information in which of these slides for you to tell which of the cells that I talked about it is. I'll even, to make your job easier, I'm just going to copy here this list all across the other slides so that you can then, remember which are the potential invariants that we might have. You ready? You got a banana? Okay, I'm gonna fry the slides. I'm gonna ask for your contribution in the chat as fast as humanly possible, okay? 3, 2, 1. Which cell is this one? You're right. That's what I like to see. Thank you, guys. Very good. Next one. Ta-da! So you can read neuroscientific data like nothing. How about this one? Ta-da! And how about this one? This one might be a little bit more challenging. As a border cell, and this is how it fires. And it's only to borders to the left of you. I don't know if you've noticed. Or actually to the right too, because the mouse goes from this direction into that direction. It talks about the border that is somehow south. Imagine that. Next one. I think that this is one that you haven't encountered as often, but it's a canonical cell. And I mean, the hardest of them all is going to come next. So how about this one? Odor. All right, guys, thank you. So ultimately, what I wanted to get across is really this idea that when you measure something, you have an empirical invariance that you want to extract from your neural processing. Our deep learning neural networks give us a very good sense of how they come to be, and we're going to come to that now. So you measure stuff with different techniques. So they're complementary, but you do have certain sort of invariances. In the case of fear, you see that the amygdala lights up in fMRI. In the case of surprise, you see that the event-related potential from EEG is a P300. So you have, you measure firing rates via a multi-electrode array in the hippocampus, and you find place cells whose function is localization. So teleological thinking, the idea that we organisms are extracting function from the environment and that the brain organizes function from the environment, is enmeshed in the idea of neural processing. And so whatever the next physicist comes to you and say, oh, there is no function, that's all anthropogenic, it's all anthropomorphizations, they don't exist. Every time that a biologist tells you, oh, you're just attributing it from your human brain to a monkey brain and a mouse brain, says like, well, but that's all we have as a measuring stick for intelligence. So the teleological argument, to me, is a subjective way of looking into these and understanding that those are necessities of life, of living, or of cognitive behavior. I'm happy to discuss this for any 15 minutes with whoever wants to stay to discuss this with me. But for the moment now, I finished the first block of the lecture, and we have 15 minutes of pause so that you can get your bearings. If you want to discuss with me the notion of teleology, you're welcome to. I'll put some music to play meanwhile.
Yes, Mario, I had a question.
Yes, please.
So I wanted to go over again the first lecture on functional topography, but I can only find the second part on Canvas.
So you mean the one from?
I think it was a week ago.
Was it was the one on topography?
Yeah, the first one.
The first lecture.
From last Wednesday, I think.
Okay, I'll recheck the YouTube. I mean, like I've process them all one after the other. I'll check in Zoom if something funky happened with that recording.
Okay. Thank you so much.
Thanks for letting me know.
Hey Mario, can I ask a question?
Of course.
It's completely unrelated to anything, but I got the opportunity to help a friend, so I thought I could ask at least.
Those are my favorites. Let me just stop the recording and you ask away.